Hi, everyone. We are very excited today to have Sam Gershman, one of my long-term collaborators, and Sam has a lot to say today. I'm very excited to have him here. Before that, and before going into introductions, just a small comment to Melanie. So Sam is going to talk about learning in single cells, but we can talk about sort of the representation, hopefully, in the discussion, because the discussion is obviously longer, and Sam has done a lot of work, and we have done some work together on this. So today, John is in transit, so he's going to take a little longer to arrive. So I've been tasked by John to sort of talk a little bit in the beginning to delay the beginning of the talk, so hopefully he can arrive. So a couple of things. First, as every session, if you're new to the Learning Salon, it's a weekly interdisciplinary forum in which we discuss bridges and contentions between biological and artificial learning. Our attendees are very interdisciplinary. We have philosophers, we have neuroscientists, we have computer scientists, we have psychologists. Sometimes we've had political philosophers and historians. So it's very exciting in that regard. The interdisciplinary discussions are sometimes unpredictable in a way that usually by the third hour, all of a sudden we enter this new space where all of us are kind of learning something we didn't anticipate to learn. So it's always been fun. We kind of subvert the traditional form of a seminar in the sense that the speaker talks for a short period of time, and we spend the most amount of time in discussion. If you have questions for clarification, there is no bad question. There is no wrong question. Please add it in the chat for clarification. But if you have a question to the speaker, please add it to the Ask a Question button just below the screen and vote on each other's questions and mention Ask for Me. If you don't mention Ask for Me and it so happens that the conversation is in a place where we can invite you, we will invite you on screen. We really encourage people from different disciplines to ask questions, and people who are junior in their research, whether students or not. We have tried to create this a kind of a judgment-free zone in a way that it's okay if you have intense disagreements. It's completely okay, and you can follow up. So, for instance, John and I every now and then have a disagreement, and then for the week ahead, we have a couple of calls and texts over that topic, and the discussion just gets more deeper, basically. So, and you can return, because we have returning guests here. You can return with questions that you've had from previous times. And we usually have a wrap-up session every now and then. Another thing I wanted to say, again, in the interest of delaying introducing Sam a little bit to buy time for John. So last session, we had a lot of discussions about representation that some people found interesting. John brought to my attention a tweet about it, because there is two different approaches to perceiving disagreement. One approach is some people feel anxious about the disagreement and want to figure out what is a way to come to an agreement. But another thing is to see disagreement as a way towards understanding. And there was a wonderful tweet that someone wrote in response to sort of my 2 cents on Twitter about how to sort of discuss philosophy in neuroscience, in interdisciplinary settings. And this was this wonderful comment. I want to make sure I say their name correctly, just to make sure. So John Michael Vore said, I just watched a replay of Learning Salon 32. Let me underscore how much happens late in the salon. Stay until the end. There is a caring collective chat and on-screen push, not toward agreement, but to understanding, which is incredible to watch and be a part of, even asynchronously. So if you see disagreement on screen, even intense disagreement, know that it's a part of a continual discussion, and it's not going to be, it's not a discomfort in the bad sense. It's a discomfort in the sense that it will move us towards maybe understanding further. And John also wrote me that he thinks that what he and I are doing with disagreeing intensely on screen and then continuing those conversations session after session is normalizing disagreements, especially between, a woman of color and him. We're both minorities. We're both part of a queer community. So he thinks it's important to also normalize disagreements towards this notion of understanding, as opposed to a push towards agreement at the cost of precision. And I think that this goes very well with something that a number of neuroscientists who are regulars at the Salon, including Adrian Fairhall, did recently. They had a look back at the sort of very largely funded brain projects, the Blue Brain and the Big Brain projects in Europe and in the US. And they look at how there were situations where, because of the lack or a lack of critical engagement with the proposal that these people were giving about these promises of simulating the entire brain, things went off. And so they have this whole issue. If you have time, take a look at it. I think it's called in vivo. It's called in silico. And there is various articles that are in this in silico, and Jovo looks like he's going to put the link there for you guys. And I think it goes towards the same idea. It's dangerous not to disagree. It's dangerous not to probe each other's arguments with care, with friendship, but intensely sometimes, because we can all get sort of a little stubborn about our ideas. Final thing to say, before I start to introduce the amazing Sam Gershman. University of North Carolina, Chapel Hill, as you know, they something happened that a lot of you might have seen on Twitter. An extremely accomplished sort of journalist who had won the Pulitzer Prize and the MacArthur Genius Award, as well as many other awards. You would know her on Twitter as Ida Bay Wells. But actually, sort of, you would see, then she's basically also the author of the 1619 Project, the book, and because of obviously her achievements, she received high recommendations for tenure at University of North Carolina, Chapel Hill. But the trustees, they were sort of conservative, right-wing, and they blocked the tenure. And a lot of people, a lot of faculty on Twitter are writing about this. The faculty at her own department wrote a letter opposing this decision. And I think that this was one of those cases where it's a little bit like, I don't know, for me, it's a little like the McCarthy era. Like, there was an ideological opposition to her in spite of all of her achievements. And this opposition came from a conservative board. And they were not her peers. So it was not based on scientific evaluation. And then there were sort of other reasons that they provided. But, you know, it's maybe some time to consider this and to think about this. There was a lot of support. And I'm just going to leave a tweet that she's thanking everyone for their support here. I hope that we can maintain academic sort of tenure, especially, and academic positions, not predicated on conservative politics, especially like this. Okay, so with these things, idea of, Joe, do you want to say something?
I wanted to, a few weeks ago, you said something about the catastrophe happening in India and recommended people donate to charities. And I just want to say, there's an ongoing catastrophe in the Middle East right now, and it's very, very sad. I think probably to everybody, no one should die. And there's lots of amazing charities. Sorry, it's very personal to me, so it's been very sad and hard this week. But there's lots of amazing charities that and nonprofits. One I know of is the Arva Institute. Their idea is the environment knows no borders, and so it's Palestinians and Israelis and Egyptians and Jordanians studying together and working together, living together, and it's a beautiful place. And I'm sure there's many, many others I don't know. So I just want to encourage everybody to support those people over there who are suffering.
Thanks for bringing that up. It's a particularly difficult conversation. So I'm from Iran. I've been under American bombs in my life. I remember what it feels like when you see buildings fall and bodies carried and like a doll left in some kind of ruin. And I, had to learn that I have actually trauma from it decades later. It's especially difficult to watch because some of the kids who died in these air raids were actually in trauma programs. These were girls from the age of sort of 8 to 11, and 11 of them died. And for me, it almost feels like I'm living a borrowed life, because I could have been one of them. And when I see them, there is this sense of, and my South African friends are especially upset at the situation. South African government actually called for UN to look into crimes against humanity by Israel. It's one of those cases where people have avoided talking about it, saying it's an uncomfortable conversation. And someone was writing recently that no amount of discomfort discussing this is the amount of discomfort that people are feeling because of the war. So because your lawmakers are actually involved in this, it's important to think about it. AOC has a clear position on this. They're trying to reduce the number of bombs sold, but let's see. Okay, on those slightly less positive notes, not slightly, catastrophically less positive notes than usual. Usually we give a non-formal and I guess a familiar introduction to people. So I'm going to introduce Sam in that way. So in terms, you probably all of you know Sam by now. He is currently I think now tenured professor at Harvard, which congratulations, that was very impressive in a very short time. But it's no surprise to anyone. Sam is extremely prolific without losing depth or sort of spread of his knowledge, which is something that I've always very much admired. Sam has an interdisciplinary outlook, is trained in philosophy, in single neuron electrophysiology, has done fMRI, has done modeling. One of his main tools that he uses is computational modeling, reinforcement learning, or Bayesian modeling. I think that his perspective on things are informed by curiosity. If you guys remember the session that we had on curiosity, and a sense of kind of novelty, which I really admire. And he's very much also informed by literature, by music, by arts. And he also made some dance movies earlier in his life, which are, I don't know if they're still on your website, but it was fun to see them in 2013 when I got to know Sam. We've had a couple of collaborations with each other, and I'm always extremely impressed by how efficient Sam is. He's very fast, and he has a very sort of rapid turnaround of anything that you send him, which is extremely impressive. And I think that his mentees are probably mentors have probably very much appreciated that, as well as his collaborators. Sam also has 2 kids and plays the piano very well. What else? Yeah, I think that all in all, you can expect to hear from someone who thinks very deeply and is very impressive, but also as a human sort of approaches life with a lot of sense of curiosity and interdisciplinarity, which I appreciate. Okay, by now we have killed 21 minutes, so I think it's time. John hopefully will show up shortly.
All right. Well, thanks so much, Ida. That was really kind of you. So shall I share my screen now?
Yes, please, go ahead.
All right.
And obviously take your time.
Yeah. Okay. All right. So what are we looking at here? We're looking at a paramecium, which is a single-celled organism about 300 microns long. And you can see that it's covered in thousands of hairy cilia that allow it to move through its aquatic environment, where it feeds on mostly bacteria and decaying plant matter. And for about 1 billion years before the emergence of multicellular life, Paramecia and other ciliates were the most sophisticated life form on the planet. And they're capable of rather remarkable behaviors. And as you can see from my title, what I'm particularly interested in is if, how, and what Paramecia can learn. And so for the last 8 months or so, I've been squatting in Florian Engert's lab, doing learning experiments on Paramecia. What you're looking at here is a video with the paramecium locations tracked in this semi-transparent pool that's made out of an agarose gel. I'm not going to actually talk about my experiments today. The results are a bit too preliminary. What I want to do actually is kind of take a step back and talk about kind of the big picture. Why am I doing this? Why is it interesting? And what theoretical issues are at stake? And as a kind of preparatory remark to that, I want to remind all of you of something that Niels Bohr said to Wolfgang Pauli when the latter introduced his nonlinear field theory. He said, "We're all agreed that your theory is crazy. The question that divides us is whether it is crazy enough to have a chance of being correct." So this kind of captures how I feel about this, because I'm actually not quite sure if I'm crazy for pursuing this or not quite crazy enough. And I'll allow you guys to judge for yourselves after hearing my talk. Okay, so where do we begin? Anyone who's taken a neurobiology class will have learned a kind of standard textbook story about learning in the brain. And it goes roughly like this. Memories are synaptic strengths, and learning is the modification of those synaptic strengths. Long-term potentiation is kind of the canonical example of this kind of synaptic learning. And it's often thought of in terms of a kind of neural implementation of Hebbian associative learning. So many of you will be familiar with Hebb's postulate that neurons that fire together wire together. And long-term potentiation is basically like the cellular analog of that. Now, although this is the textbook view, there are some major problems with this view. And it's nicely summarized in this table from this paper by Gallistel and Matzel. And I'm not going to go through this whole table, but you can go and look at this paper if you're interested. And the fundamental problem that they're pointing out here is that the properties of what they call Hebbian LTP don't really line up very well with the properties of associative learning. Now, I want to say immediately that although they label this column associative learning, we have to be careful, because the term associative learning, like so many other things in psychology and neuroscience, designates both a set of empirical phenomena and a theoretical explanation of those empirical phenomena. And if you ask Randy Gallistel, he will gleefully tell you that no animal on Earth has ever formed an association in its brain. So he very much disavows the idea that animals are actually learning associations. But what this is taken here to be designating is a set of phenomena, a set of empirical regularities about learning. And let me just give you one example from this table. Okay, so when we talk about neurons that fire together, that wire together, there's an implication there about simultaneity of firing. But of course, nothing happens exactly simultaneously in the brain. So what we're really looking for is neurons that fire close together in time. And so the more nuanced version of Hebb's postulate in the brain is spike timing dependent plasticity. And the basic result there is that if the postsynaptic neuron fires within a short time window after the presynaptic neuron fires, then there's long-term potentiation. And if it fires in a short time window before the presynaptic spike, then there's long-term depression, so actually a weakening of the synaptic strength. Now, how short is that window? Well, you can see that right here on this plot. It's about 40 milliseconds, and maybe even shorter over here on this side. Okay, now, how does that line up with the timescales of behavioral learning? Now, it is true that if you increase the interval between a stimulus and an outcome, like a conditioned stimulus and an unconditioned stimulus in a classical conditioning experiment, then learning will take a longer time. And that's shown in this plot in this black line. So this is the interstimulus interval. And this is trials to acquisition. So it's the number of trials you need to meet some acquisition criterion. And you can see that this is a monotonically increasing function. So it says that as the delay between these two events gets longer, learning a conditioned response will become slower. Okay, so that fits with this broadly speaking, although you'll immediately see that like for spike timing dependent plasticity, nothing is going to happen after 40 milliseconds, more or less. But this delay is measured in seconds. Now, even more critically, actually, is that this is a result where you hold the intertrial interval fixed. But if you allow the intertrial interval to be adjusted such that the ratio between the intertrial interval and the interstimulus interval is constant, then the trials to acquisition is basically constant as a function of the interstimulus interval. In other words, I can learn the same amount from whatever delay you give me, as long as you space the trials out proportionally. Right, so that's a really remarkable result. It's sometimes referred to as the timescale invariance of learning, and so it indicates a kind of radical departure from the extremely stringent temporal requirements of synaptic strengthening and weakening. Here's another problem for synaptic plasticity. Synapses are not really very durable entities. So, for example, in auditory cortex, 60% of dendritic spines are replaced within 3 weeks. This was something actually that several decades ago Francis Crick was aware of, and he wrote a short note about this. And he pointed out that the timescale of molecular turnover is much faster than the timescale of memory. So not only are the dendritic spines being replaced, but also receptors on the postsynaptic membrane are getting removed and degraded. So the thing that we call synapse is really a quite unstable entity. Now, that's not necessarily a deal breaker for the synaptic plasticity hypothesis, but you have to invoke other kinds of mechanisms, typically at the circuit level, to compensate for that. All right, so now I want to go from kind of relatively well-known problems with synaptic plasticity to more wild territory, okay? So here are planarian flatworms. And planarians have amazing regenerative capabilities. You can cut off 99% of their body and they will grow back. Now, here's a question. When you cut off a planarian's head, it will grow back. What, if anything, is retained in that head? So it turns out that planarians are capable of classical conditioning. This was shown by James McConnell in the 1950s. And so he did, in particular, tone shock conditioning. Now, if you cut off the planarian's head, it will regrow, and that regrown head will retain memory of the tone shock association. And this was replicated more recently by Schoenrath and Levin. Now, how does one think about this in terms of synaptic plasticity? All the synapses are being destroyed in this experiment. So something else has to be going on, if you believe it. Now, even wilder and also much more controversial is this claim that you can transfer memories from one planarian to another by feeding, so that they will actually engage in cannibalism. They'll eat each other. So, the simplest experiment that McConnell did is he just chopped up the trained planarian and fed it to an untrained planarian and showed that he could transfer the memory. And then later experiments refined this to purify RNA from the organism and then inject it into another planarian and show that that could also transfer memories. Although this was extremely controversial at the time because of various replication failures. But many other labs showed an analogous phenomenon, not just in planarians, but in other species as well. For example, in rodents, in Apparatus, in goldfish, recently in C. elegans. So I think that there's something there, right? I don't think memory transfer is complete nonsense, even if some of those experiments couldn't replicate. And we can maybe later we can talk about why those experiments maybe didn't replicate. Partly it's because, in some sense, the psychologists and biologists who were working on this at the time were kind of thinking far ahead of what they could feasibly study with the tools at the time in the 1960s. So there was this hypothesis that memories might be stored by macromolecules like RNA. And that was what motivated them to try purifying RNA and transferring it to another organism. but they kind of that hypothesis kind of died out with the memory transfer experiments and was eventually replaced by the kinds of textbook stories that I told you before. And I'll come back in a minute to talking a little bit about RNA. Here's another interesting example. So there are many species that undergo quite substantial brain remodeling during their life cycle. So for example, when a caterpillar turns into a butterfly, so these are kind of metamorphosizing organisms. And then there's also organisms that hibernate. Hibernation induces quite radical brain remodeling. So synapses, many, if not most synapses get replaced by these processes. And yet studies in the wild have shown that these organisms retain information prior to metamorphosis or hibernation. So for example, butterflies can retain olfactory memory of larval environment. These Arctic ground squirrels can retain kin recognition and retention of a trained task even. Okay? So again, the question is, if you accept a synaptic plasticity hypothesis about memory, how do you explain retention of memory across dramatic remodeling of synapses? Now, we could take this even further, and this is kind of where I ended up, which is, why even bother with synapses? Why not study organisms for which synaptic plasticity just isn't even an option? And that's why I converged on Paramecia. And another reason for this is because there is a literature, much of which was really forgotten about, on learning in Paramecia. And so, for example, there are these studies by Beatrice Gelber that showed that Paramecia seemed to be able to associate platinum wire with bacteria, which is a food reward for them. So this is a schematic of these experiments that Gelber did. It's a beautiful illustration from this article in The Scientist, which I think was shared with all of you. And we discussed this at length in this retrospective piece that we wrote for eLife. So basically, I'm not going to go into all of the different variations of this experiment. She did quite a few experiments, but basically she would coat the wire with bacteria and put it in this dish where the Paramecia had been pipetted. And then the Paramecia would basically glom onto this wire. And the question was, what happens when I test with no bacteria on it? The Paramecia still glom onto the wire. And that was considered a form of associative learning. And just as a kind of meta-scientific point or kind of sociological point, at the time, not that many people accepted Gelber's results, and there were some fairly vociferous objections on various fronts, and Gelber tried to meet many of them. She was basically this kind of lone champion of this idea. Really, no one else was working on it at the time. And unfortunately, the conclusion that people drew from that episode in scientific history, much like for memory transfer, is that basically it was not real. The effects were not real. They had alternative explanations, confounds, and so on. And so the subsequent reviews on the topic would basically say, yeah, Gelber did these experiments, but like they weren't right for various reasons. And so that was the reason we wrote this retrospective, is because we wanted to revisit that conclusion and try to kind of pry open the history books a little bit and allow a different perspective in. I'm not saying that, I think that basically there were issues with Gelber's experiments, but I think that she was onto something. I think she was studying a real phenomena. There's another interesting paper, and this was really kind of disappointingly a one-off, by Todd Hennessy and colleagues, where they were looking at classical conditioning in Paramecia with a different setup, which is similar to what I've been doing now, where they used as the CS a tone, which essentially for the Paramecia is a vibration. And then for the US, they used an electrical field. So it's known that Paramecia will reorient it and swim towards the cathode when placed in an electrical field. And they did a whole bunch of really interesting experiments. For example, they showed that Paramecia learned this, they retained the memory for up to 24 hours later. They had very nice control conditions like scoreless, truly random control, where the CS and US were still presented, but their timings were randomized, so there was no temporal contingency between the events, and the Paramecia didn't show an acquisition of conditional response in this random control condition, but did when they were temporally contingent. Okay, so if Paramecia don't have synapses, how do they store information? That's kind of the million-dollar question. And there are a bunch of possibilities. I'm going to mention three prominent hypotheses here, and then I'll finish up shortly. So one idea, and this goes back to ideas from the 1940s and 50s, is that memory is stored in a polynucleotide sequence or some other micromolecule. So for example, Galstel has argued strongly that it could be RNA. And he has various first principles arguments for why that should be the case. RNA is thermodynamically stable. It's energetically cheap to write and read nucleotides. And more recent data has suggested that there are ancient mechanisms in cells for packaging RNA and sending them through the extracellular space in vesicles to other cells, which can then unwrap them and read the contents, and that actually has behavioral implications. So it's not just an exotic hypothesis. My favorite recent example is work from Colleen Murphy's lab, which showed that C. elegans will acquire taste aversion when they eat some toxic bacteria, and that taste aversion memory can be transmitted to both vertically and horizontally, so to other worms that can ingest these capsids, these packages of RNA that were produced in response to the toxicity event, and also it's heritable. And she actually points out that this happens in nature. So if C. elegans dies from this toxicity, they will sometimes lyse and basically burst open, and then other C. elegans will consume the remains, and so that way they can actually acquire these memories through these RNA packages. Two other important possibilities are DNA methylation and histone modification, and I won't go into the details of this, but basically these are nuclear changes. So DNA methylation involves adding methyl groups to DNA. Histone modification involves methylating or acetylating the chromatin that packages the DNA, so kind of the wrapping paper for DNA. These have all been implicated in memory in various ways. And interestingly, they've also have a long history in cell biology in the study of epigenetics. So these can also be heritable changes. Now, I don't know how many neurobiologists are in the audience right now, but there's I think a standard response to what I'm talking about right now is that, well, yes, of course, these things are involved in memory, particularly DNA methylation and histone modification, which have been studied quite extensively in neurobiology, but they're not the memory substrates. Memories are not being stored in methylation patterns or histone patterns. Those are just mediators of synaptic plasticity, because we know that nuclear events have to occur in order for synaptic plasticity to occur, right? So there's a whole pathway by which spiking and calcium influx will set in motion an intracellular signaling cascade that results in gene expression and ultimately protein synthesis and trafficking of new receptors to the postsynaptic membrane. So maybe these are all just mediators. But I guess the challenge that I would put to people who adhere to that view is, how do you explain all the things that I basically just said, right? Like, how do you explain memory transfer? How do you explain memory following brain remodeling? How do you explain memory in organisms that lack synapses entirely? Now, there's also a reverse challenge, and this is the last thing that I'll talk about, which is, all right, so we know that synaptic plasticity happens, right? So if it's not storing memories, what is the function of synaptic plasticity? And here I want to call attention to an idea, and this is really total conjecture, okay? So I'm happy to discuss it, but I don't have any direct evidence for it, which is that there are two kinds of learning. And this comes out of work that my lab has done studying cognition in humans. So there are facts about the world, like, where did I park my car? What time is dinner? And then there are computations. Okay, so if I learn facts about the world, I still need to access them and make inferences about hidden variables, sometimes conditional on other facts. So I have to really solve two problems. I have to learn a generative model of the world that encodes the various facts that I know, and then I also have to learn a representation that allows me to compute with that generative model, to actually answer queries. And this is sometimes called a recognition model. Here I'll appeal to some important developments in probabilistic machine learning. They really started several decades ago with the Helmholtz machine, which was developed by Peter Dahan, Jeff Hinton, and colleagues, which combined learning of a generative model with learning of a recognition model. So the idea there was that if you have to do inference, you can build an inference engine that's basically a parameterized function, like a feed-forward neural network. And now you can simultaneously learn the parameters of your generative model and learn the parameters of your recognition model. So learning the generative models, learning facts, learning the parameters of the recognition model is learning to infer. And we've done a bunch of experiments, which I won't go into, with humans to basically argue that humans are doing this kind of learning to infer. And so I want to put out the kind of tentative thesis that facts are stored in a durable molecular format within the cell. But in multicellular organisms, which encode lots and lots of facts, they have to now solve the problem of routing computations to take some facts, combine them with other facts, make inferences about unobserved facts, and so on, right? And that requires intercellular communication. And synaptic plasticity is the mechanism by which the parameters of the recognition model is learned. That's my hypothesis. All right, I'll stop there and we can have a discussion.
Thank you so much. There was a fun chat going on and there are some questions, but I think that you did a great job of explaining. So I'll ask for some clarification questions just to remind everyone of the three sort of, you mentioned three hypotheses. So in short, how would you, if you wanted to say those three hypotheses and what your opinion is on which one in your opinion makes the most sense, and maybe like very briefly, like a sentence each, just as a mnemonic device, so that everybody sort of remembers.
Right. The three.
Last.
Should I? All right. I'll just describe it. So the three hypotheses, and this is not meant to be exhaustive, are memories could be stored in the form of a macromolecule like an RNA. Memories could be stored in a pattern of DNA methylation or in the pattern of histone modifications. By the way, with regard to the RNA hypothesis, I want to just plug some amazing work by Hissam Akhlaqpour. I would never know if I'm pronouncing his last name right. But he wrote a paper, which I'm sure someone will inevitably link in the chat, on universal computation with RNA. So he showed that you could actually implement a universal computing system just with cleavage and ligation of nucleotides in an RNA sequence. So that kind of lends plausibility to the idea that RNAs could be kind of fundamental computing units and possibly store memories. I'll mention one other thing. Sorry, I don't want to take up too much oxygen here. So David Glansman, whose lab pioneered memory transfer in aplesia, thinks that RNA plays a temporary role in memory storage, but ultimately he thinks that memories are stored in nuclear format, and his preferred hypothesis is DNA methylation. So it's possible that RNAs are maybe playing a temporary role, but not for long-term storage.
So this somehow brings me to, okay, so maybe just to make sure that the clarification is there, because not a lot of our audience is actually familiar with genetics. We have philosophers, neuroscientists, computational neuroscientists. So what is sort of the significance of the difference between the RNA versus the DNA methylation versus the modification hypothesis? So what would be the consequence if you could frame it in a sort of a bigger picture, both maybe in terms of the lifespan of the individual? You mentioned that RNA might be more temporary, as well as like what would be the key theoretical difference between these three that you could say, oh, you know, here is the predictable experiments that each of them can do.
So I think it's a really hard question to answer, but a really important but very hard question to answer, because we really just don't know. It's actually it was quite disappointing to me in a way in reading this literature, and I admit that I'm very much still a novice. That there's a whole literature on what's called the histone code, right? So I said, oh, histone code, that sounds great. So they figured out what the code is. No, that's just a metaphor. Nobody knows what the histone code is. What they know is that histones are somehow involved in storing information about cellular identity, that it's modified by experience, that it's capable of being inherited. So it's a mechanism for epigenetic transmission. But what's missing, right? If you say you know the code, what does that mean? It means you know the code book, right? It means you know how to translate between some information that's being whatever it might be, you know, some protein that that could be expressed by a gene, and the underlying biological substrate, right? So when we say that we know what DNA codes for, we say that it codes for a protein, right? That we can basically read out from the genetic code what proteins are going to be. I know that's a little bit of a caricature, but we don't have the analogous understanding of things like chromatin, the histones, or DNA methylation patterns. We know that in principle they're capable of storing a vast amount of information, but we don't understand what the code book is, right? If you want to talk about representations, we don't know what they represent. And I would say the same thing. I just would make a meta point here, which is I think this problem ubiquitously plagues discussions of memory in the brain, because we're constantly talking about memory in the brain, but no one except maybe Randy Gallistel is ever talking about the content of memory in the brain, right? Because we're so fixated on an idea of memory as associations, which are not by themselves representational units, that it's very hard for us to discuss like what is represented by a particular biological entity, right? So from Gallistel's perspective, for example, the brain needs to represent things like numbers and places and times. And so in his view, there has to be an explicit representation, and he thinks that explicit representation is in the cell. And so if you take that idea seriously, then what you want to look for in the cell is like an RNA sequence that encodes a particular time interval or location, right? But no one does experiments, the kinds of experiments that you need to do to establish that kind of conclusion, right? Because no one is saying like basically it's kind of like we're in can a biologist fix a radio territory. Like you futz with the DNA methylation and say, oh look, the rat can't store memories anymore. But you can't say like, all right, what is this particular methylation pattern encode about the rat's experience? That we don't know. All right, I'll stop there.
Thanks, that's very helpful. Also, Bahar Ghalipur, who's one of our, first I linked Hessam Akhlaqpour's paper that you mentioned. And the title of the paper is An RNA-Based Theory of Natural Universal Computation. And Bahar Ghalipur also, who's helping us at the Salon, added that histones can flexibly carry flexible marks that say which gene can be accessed. Okay, people are writing things and my chat is moving.
Bahar.
can't see anything anymore. Access for transcription right now and which can't. Basically a way to make sure every gene gets used precisely the way it should or at the right time. So changing histone marks, methylation, etc., changes the story that the DNA code can tell at any time, which is a wonderful way to write it. So I really appreciate what you explained, and I think that once I ask my questions, you'll see which trajectory I'm taking. What's the sort of with these clarification questions. So just one thing about one last thing, if you could explore, is there a difference in the time scale of these three different hypotheses?
That's great. Great question. I'm not sure because one thought is that the reason to have kind of temporary transmission with RNA and long-term nuclear changes is that you can modify RNA quite quickly relative to changes at the nucleus. But to be honest, I'm not actually sure how fast things can happen. Maybe someone in the audience knows.
So so given that the memory is being transferred when somebody eats this creature, like how long that memory lasts? I'm a worm. I just eat another worm who had a memory of being very averse to the smell of apples. How long am I going to not like apples?
Gosh, that is a great question, and I wish I knew the answer to that, because I'm not sure if I wouldn't be surprised if Colleen Murphy's lab addressed this question, but I just don't know the answer off the top of my head. But one thing that they also showed is that transgenerationally, this memory can last for 4 generations.
That's crazy.
Yeah.
Here is the reason I'm asking all of this, and I think it's very exciting. It's the first time I'm actually thinking about this, but it connects something that I'm doing with meta learning with what you were describing right now. So I wonder if these different 3 hypotheses work at different scales because. So imagine that there are some memories that I need for my life. Imagine a base learner and a meta learner. The meta learner works at the level of maybe generations, maybe even evolution to another species. If you mess with the histones, you probably are going at that meta learner level. You're not just thinking about one entity, if that's the level you're working on. The RNA, I'm not sure. It could be also shorter scale, shorter time scales, like you were saying, maybe more temporary. So what I'm trying to think now is how the way that memory can link things that you learn within one lifetime and things that a species might learn within different lifetimes, and the ways in which DNA changes as a way of, a meta-learning perhaps that one could imagine is happening across evolution, across even different species. Because in a manner of speaking, DNA is a kind of memory. It's a kind of learning, and evolution is a kind of learning.
Yeah.
Not a monotonically more learning, but just in different directions of learning. The way that we have different disciplines, maybe, and the ways in which some experiences turn into changes that might change the DNA, or might change across First, it might change me, the way I react as a one individual of that species. The way it might act, like you were saying, across generations to my offspring. And third, the way it might change my species or evolve into the next. So I wonder whether you think there is a connection between these very different scales when you're looking at the RNA or DNA level of change due to memory. I wonder whether there is like a thread that is linking this individual level to that sort of higher level, and it's just different in higher sort of as in more cellular, multicellular species as opposed to single cell species.
Yeah. Have you guys had Tony Zader talk about his views about this yet? Okay, so I mean, I kind of like his take on this. I wouldn't say that it's particularly controversial. I think like part of the confusion arises when we try to take these machine learning paradigms for meta learning and attach them to biology, because it's just very unclear what the mappings are, and so there's a lot of hand waving, like, when I train this network, I'm doing kind of both evolution and development at the same time. And it's not really clear where the boundaries of one ends and the next begins. So
one comment on that.
Yeah.
So the meta-learning I'm talking to you about is not within a network. This is actually species and generations.
Right. So no, I understand. Right. Yeah. So you're describing like meta-learning as it occurs in biology, right? Which is evolution. I mean, right. I don't think anything I said contradicts the idea that there's learning at evolutionary timescales and developmental timescales. I mean, yeah, I guess I'm not sure what to say about that, apart from just that, like, both of these things are happening. And the question in my mind is whether we've basically been neglecting a form of learning at the developmental time scale by focusing too much on synaptic plasticity. Right?
Oh, absolutely. I was just taking it even a step further. I was saying it might even link these two different kinds of learning, the developmental kind of learning and within lifetime learning and the kind of cross-lifetime learning. So that's the further sort of
oh, I see. So right.
To make your idea crazier.
Right, because basically, like, if we're talking about nuclear changes, you're actually modifying the kind of genetic stuff, right? So now it blurs things again, because yeah, of course, like, just any epigenetic argument is going to blur these lines. Right.
But in this case, you might have these three hypotheses might differently give rise to this. So that's the reason I was asking about the time scale. How long does each of them last? Which one can be transferred? And you mentioned one of them is 4 generations. One of them is more temporary. So all of the above. So John says that he will be here shortly. Hopefully that is going to work. He's off the plane. So yeah, because we have a lot of questions and not a lot of time, I'm going to ask Jovo to ask his questions before we go to the audience.
Cool, thanks, Ida. Can everybody hear me? Okay, great. So Sam, that was really cool. You know, I worked on synapses for years with Stephen Smith, and it was just, I love the like summary and history. I learned so much in that 10 minutes, even having worked on synapses for like 10 years. So that was cool. I wanted to ask you just some basic clarification questions. You've talked a lot about learning and memory, and you define learning in the Eli paper I read in advance, but I wanted you to say it here. Like, when you say learning and or memory, can you just define it for all of us? Like, what do you mean?
The broadest possible definition of learning is any modification as a function of experience. That now you can qualify that in various ways. Like, it has to be sort of more persistent, and sometimes an additional qualifier that's invoked is that it has to be functional. Like, you know, if I bump into a wall and then like walk the other direction, right? Is that a memory, right? It modified my behavior from my experience, but I'm not really storing some information necessarily that I'm going to retrieve later. Now, what about like if someone cuts off my arm? Well, that is a durable. That's a persistent modification as a function of my experience. But would we call an injury a memory? We probably would not want to. So that's why these qualifiers of persistence and functionality are sometimes invoked, but they're very slippery, right? Because how long is persistent? Like, obviously we forget things, right? So even things that we would all agree are memories can have some lifetime of retrievability. And likewise, functionality, who's to say what's functional? Like, we don't really know. We're often in this situation of sort of, we know it when we see it, but it's still useful to kind of work through what we mean by these definitions. I'll also say that that biologists are often very uncomfortable with imputing learning to various organisms like plants or single-celled organisms or slime molds, and are more comfortable with the term adaptation. Sorry. So then we're in this semantic zone where like, what is the difference between adaptation and learning? I'm not sure. I think it is Audrey de Sautour is maybe in the audience and has an opinion about that. But I don't know. So to my mind, as a psychologist, I'm not so worried about the distinction between adaptation and learning.
Okay. So I have some follow-up questions just to see. I agree it's clearly slippery and maybe it doesn't matter that much where the boundaries are, but I just want to get a better understanding of where you stand. So you talked a lot about single-cell organisms learning, but those mechanisms are presumably preserved in multicellular organisms. So do you think that single cells within organisms are also learning?
Well, yes. I mean, that, okay, so thank you. This is something that I didn't quite get into, but it's another reason why Paramecia are interesting as a model system, because Paramecium are actually nicknamed swimming neurons. Why is that? Because there are very influential electrophysiology studies in the 1970s and 80s, which showed, for example, that Paramecia have action potentials, and those action potentials control voltage-gated calcium channels that in turn control cilia movement, among other things. They have intracellular signaling cascades that are highly preserved across all eukaryotes. So for example, calmodulin is an important calcium signaling pathway. Now, that kind of raises the plausibility that whatever mechanism we might discover for learning in Paramecia might be conserved in other organisms and more complex multicellular organisms either. Now that's total speculation at this point, right? But it from just a kind of molecular biology perspective, it's not totally crazy.
Okay, and then you focused a lot on single cells, but of course there's many different spatial resolutions, and so I wonder, do you think, assuming the synapse story is true, do you think an individual synapse can learn?
Wait, sorry, do I think an individual synapse can learn? Well, yes. I mean, so nothing that I said should be construed as denying the existence of synaptic plasticity. What I'm trying to do here. Yeah.
I just mean not synapses used in the process of an organism learning, but rather the synapse itself has learned as an entity unto itself.
Wait, how is that different?
Like organisms can learn, cells can learn, how low can, like I think civilizations can learn. There's lots of scales. Where does it stop? Like cells learn, I'm with you. The synapses learn, the molecules learn. What?
Well, okay, I mean, maybe we're venturing into a philosophical territory of what constitutes a thing such that learning can be a property of that thing, right? You know, can rocks learn? When I throw a rock down a hill, I've modified it on the basis of its experience, but you know, that presupposes that rocks can have experience. So, I mean, I don't know. I mean, I'm not sure that it matters for the purposes of the kind of questions that I'm asking, which is that we take for granted that there are these organisms that are capable of sophisticated behavior, and you could in principle say that they learn. It's still controversial. And if they do learn, how do they learn? And does that have any implication for how more complex organisms learn? And we can address those questions without answering the question, how low can we go? Even though I agree that's an interesting question, but maybe not the one that I'm trying to answer.
Yeah, I think, you know, the reason I ask you is because I'm interested in understanding the bridge between natural and machine learning. And so if rocks can't learn, I wonder if machines can learn.
Oh, okay, I see what you're driving at. Okay. Yeah. So no. I'm not a vitalist, right? So I absolutely believe that computers and machines can learn. So I think that the relevant question is, you know, what mechanism do you have to build such that any kind of machine or agent or organism can learn, right?
Okay, I'm trying to let John in. I'm failing, and I'm not entirely sure why. I will keep trying. In the meantime, just as one more question that I posed in the questions. So it seemed like you thought maybe that worms can eat memories. I wonder if you think humans can eat memories also.
Well, I don't know. Why not? But I haven't seen it demonstrated. I mean, the thing about the C. elegans, right, is that it's you can make the argument that they were maybe evolved to do this, right? Because there's clearly things that they need to learn that could be transmitted in this way, right? Like taste aversion could be transmitted in this way, possibly. So maybe humans can transmit taste aversion in that way, but I don't know. We don't normally eat each other and consume our RNA capsids.
Not anymore. Thank you for placating my question.
No, thanks. Those are interesting.
So we have a lot of questions, and while Jovo is going to struggle with inviting John and Rosa, hopefully she can also join us. It would be fun to hear her perspective. I'm just going to read maybe some of the questions. And I have so much more to ask. I hope that you can stay a little longer than you thought. Yeah, what about plants? I think that's a great question, John. So there are many interesting questions. I'm just going to read maybe the ones that are the closest to what has been already discussed, so it might have a fast reply. So should DNA mutations be classified as memories? Yuri asked this.
So which mutation? I'm just trying to find where this. Which mutation?
Should DNA mutations be classified as memories?
Well, it's interesting because, I mean, well, first of all, just on first principles, why not, right? Like, I guess if someone asks the question, is this a memory? Like, I guess I would ask, why not? Why couldn't it be a memory? If it's a persistent modification on the basis of experience, why not? But I mean, I think again, if we're concerned with if we think that cells are storing certain kinds of memories, we'd like to understand how those memories are stored. What is the format? How are they stored and read out? So then it's an empirical question whether DNA mutations correspond to that kind of the modifications necessary to store those kinds of memories. So we always have to just keep in mind what kind of memories are we talking about.
So Montreal has a related question. I hope you can invite Montreal, Jovo, while I read another one. So so Elle asks, can we say that shape memory alloys have memory? Metals that can be deformed but will go back to their original shape given specific temperature conditions. If not, what would be simplest example of a memory, and is the difference that it has to be in a biological organism? Maybe we skip this one.
Well, can I just say one thing about that? Because I was pondering this a few weeks ago. You know, like, is there some pan-computational definition of memory? We could say the whole universe is a memory, right? And I think that one way to answer this question is that biological systems have evolved timescales of memory that are specifically useful to them, right? That promote fitness. So organisms have to remember things over certain timescales and then retrieve that later and act upon it. And different organisms will need memory to be conserved over different timescales potentially. And some organisms will need memories over multiple, vastly different timescales. So I think one big difference is that, like the question I was thinking of was, when you look up in the night sky, you see stars that are memories of things that happened, you know, millions, billions, trillions of light years ago. So you could call those memories, right? But an organism has to those memories are not necessarily useful for life on Earth, right? We're not storing information at that time scale for retrieval later, right? So I think that's the key difference. You know, a physical material could store a memory, but we as organisms have actually engineered it to store memories at time scales that are useful to us.
Maybe it's time to go to the folks on screen now before we read more questions. So Montreal, do you want to ask your question first? You had a highly voted question. You are muted if you're talking.
Am I still muted?
No, now we hear you.
Okay. All right. So one, I want to say thanks for the presentation. It was really cool. It reminds me of my molecular genetics class that I took, which is really fun. So before I ask my question, I want more clarification on two things. One, at first I thought you were going to get at like learning, so behaviorally, cognitively, but it seems to me like you're more interested in the phenomenon of learning in itself and how it can be captured either at the synaptic level or molecular level. And then two, are you saying learning is simply memory transfer that you can probably identify either?
No, yeah. Let me clarify. So I'm definitely interested in the cognition of memory, but for the purposes of this talk, I'm focusing on kind of the general biological principles that would be needed to support storage of memory in a format that's actually useful for cognition. So what I was describing when I talked about Galstel's arguments against Hebbian LTP, in those arguments, he's very focused on the fact that LTP or STDP, they don't have the properties that you would need, at least on their own, to support the cognitive information processing functions of memory. That's his argument. And in answer to your second question, no, memory is not just memory transfer. But what's interesting about memory transfer is how is it possible? What format do you need to store memories in that for which memory transfer would be possible? It does not seem obviously possible if you think memories are stored at synapses. But it does seem more possible, and indeed has been empirically demonstrated that if memory is stored in RNA molecules, then you could package those and transmit those to other cells, and that might even be something that happens in vivo.
I see, right, I see. Yeah. Yeah, so I'll ask my question, which was, what do you think, so as you identify and sort of characterize the molecular storage of these memories and potentially how they may or may not be the learning mechanism, I guess, it's the one interpretation. What does that tell me potentially about more at like a behavioral level or skill learning? And I think the example I used was the sort of histone modifications, right? Because you can phosphorylate or dephosphorylate and that changes its activity. Mm-hmm. Can that tell you or give you some insight into either what occurs at skill learning or how we can approach studying skill learning, I guess?
Sorry, studying, you said skill learning?
Skill learning, yes.
Yeah, skill learning.
Yeah,
well,
which I should say that the assumption is that, when you learn a skill, you become sort of, not skill learning, I'm sorry, adaptation, you become adaptable. You sort of use your lookup table, if you will, in different ways.
Yeah. Yeah, there are many ways to answer this question. It's a good question. And I should again add the caveat that this is all quite conjectural. If you think that skill learning consists at least partly in storing things like numbers, places, the kinds of facts that Galstel likes to talk about, then you need a format for storing that information that has various properties. So it needs to be robust to degradation. Okay, so if you accept that synaptic plasticity, synapses are not particularly robust to degradation, then that could potentially be a problem. It should be metabolically cheap if possible, to both read and write. Spiking activity is enormously metabolically expensive, as is protein synthesis for constructing synapses. Whereas modifying polynucleotide sequences is extremely metabolically cheap, and they're thermodynamically stable. You can preserve DNA for hundreds of thousands of years. So and then the other thing is it has to be scalable, right? So if you have to encode time intervals, for example, over scales that differ by orders of magnitude, you don't want like naive labeled lines, which will exhaust their capacity once you reach a particular order of magnitude. And here again, I'm being sort of a mouthpiece for Gallistel. You know, he points out that that people and computer engineers have solved this problem to some extent, right? Because we have digital computers and we have coding schemes that allow us to encode vastly different orders of magnitude in the same digital machinery. And so that's why he was attracted to polynucleotide sequences, because he thought that plausibly it could be a kind of a digital code for things like time intervals. But histone patterns, DNA methylation patterns, those have also been analyzed as digital codes. So they could also subserve the same function.
Great. Oh, right. So thanks. So I have to like.
Sure, yeah, go for it.
Thank you.
Thank you, Montel.
Yeah, of course.
Hi, John and Rosa. John, we're happy to have you, and I guess we will wait for Rosa to share because you just joined, so hopefully you'll capture. Or are you unmuting? Or no? I don't know where you are. Are you trying to unmute? We can't hear you.
I am, yeah.
Okay, now we can't see you, but we hear you. Okay. John, we can still hear you, just FYI.
Can you hear me now?
Yes, and we can see you.
You can hear me, right?
Yes.
Oh, okay. Well, anyway, hello, Sam. Sorry, I feel like I've been really rude. But Rosa, you go ahead and ask.
I don't know what specific question you wanted me to ask, but I just had a thought on what Sam was just saying. It seems like there's an important difference between single-celled organisms and creatures like ourselves in this case. If you're thinking about storing information in polynucleotide sequences, if you're a single-celled organism, maybe that basically works the same way as storing things in molecules, in other kinds of molecules, right? Like it's very easy to read it out. There's only one cell and it's like directly hooked up to the action/motor producing machinery.
Well,
it seems like in a multi-celled,
right?
Sorry?
I wouldn't say easy. I mean, chemical computers are really complicated, but anyway, sorry, go on.
You can see how it would go, at least in principle. And it seems like in a multi-celled organism where there are like quite specific groups of cells that drive behavior and that we think are responsible for all the other cognitive stuff that memories have to interact with, it would be quite difficult to figure out which cell, you know, the relevant DNA methylation marks or like histone modifications were stored in. Either you'd have to have them in all of them, or you would have to have some way of reading out just the particular ones that had the right modifications. And I think you have all the same problems that Gallistel thinks happen with synapses again, right? Like it's really hard to read out information that's stored in those ways. It's slow. It's probably energetically expensive because how do you get any outside effect from making a small change in a cell internal RNA?
Yeah, I mean, it's a challenging problem, and I'm not going to claim that I have the answers to this. I think, I mean, going back to the last thing that I said in my presentation, the idea that I'm interested in now is that in multicellular systems, there's a process of learning to infer that's mediated by synaptic plasticity, and that's basically the process of learning to kind of route information from one cell to another, even though the information itself is being stored in a durable format inside the cells. Right? So I mean, I have no idea whether that will truly solve all of these problems. I mean, someone has to build an explicit model of how it works. But I guess I don't see why, in principle, it couldn't. But maybe I'm missing an angle here.
Well, I should say I was not actually able to make your talk, so I don't know what you said. And
Okay.
so I probably
Sorry.
Shouldn't be commenting on anything except what I just heard you say.
Okay. Yeah. I think it's an interesting and important problem. But for the paramecia, right, with they're only one cell, right, as you say, like we can kind of sidestep that a little bit.
Just
Okay,
a couple of things.
Go ahead.
Sorry, go ahead. No, continue.
Well, I don't know if you said anything about this in your talk, but like it is genuinely puzzling to me how these memories are supposed to manifest themselves if they're stored in this cell internal way. And it seems like it's kind of. Sorry,
I didn't mean to cut you off. Yeah. Go ahead, please. Yeah.
I mean, it seems like it's important to someone like Galistel's conception of memory that it's something that can be read and written. So maybe you have some story about how it gets written, but it's very weird to think about how information like that gets read out, right? So if all you're storing is the quote memory of a behavioral disposition, so like anything that consumes this is now more likely to produce a certain kind of behavior, you can call that a memory. But I think you could also just call that a physiological modification that you don't have to assign any kind of content to, right? It's not a memory about anything. It's just this disposition to behave differently. Whereas for something to really be a memory, I think you want it to be able to drive like a range of different dispositions in different contexts. Like that's what makes us want to say it's a memory, it's about something, as opposed to just we changed the thing somehow by cutting off its arm or pumping it full of ATP or whatever it is.
Wait, so aversion to a particular taste would not be considered in the domain of learning and memory for you?
I think it depends, right? So it depends on whether it's a preference for a particular taste that could be expressed in a range of circumstances, for example, that could be expressed by various different kinds of behaviors. If you're just changing like a single behavior, then it seems like you could explain that without saying there was a whole memory responsible for it. So I think, I don't know if you guys talked about this, but I think this comes up in like the inception experiments a little bit with the optogenetic engram plantation from the Tonagawa lab. Like there's a thing where like the behavior changes, but it's like a one-dimensional thing, right? It doesn't seem like it's enough to allow you to attribute like a whole episodic like memory to the thing.
So
that's sort of a digression, but the point is like it seems like what matters for something being a memory is that it doesn't just change a single way that the organism might behave. Right? It has to interface more thoroughly with the rest of the cognitive apparatus.
So in single cells, I'm not sure if we can talk about a cognitive apparatus.
Sure. Yeah.
So I think that it might be a different scenario if we're talking about like multi-cell or mammalian kind of type of memory, or in this case, single cell. But I'd love to hear what Sam is saying.
Yeah, I mean, I think you can talk about a cognitive apparatus. I think we're so afraid of talking about cognition in these lower organisms, but I guess I'm not. I am less afraid, I suppose, for better or worse. But I don't know if I'm not sure if I agree with the idea that you have to have representational content to be a memory, as opposed to a disposition. I worry that if we define memory so narrowly, we're going to exclude things that we might want to call memories. But however that may be, people like Galistel, and I'm kind of in the same boat, I think that our attitude is that there are representational memories, whether or not there are other kinds of memories, and we'd like to and there's a strong argument to be made that there are such memories in multicellular organisms, and I'd like to understand whether there are such memories in single-celled organisms. Oh, and sorry, and just to answer the reading question, right? I mean, we have intracellular mechanisms for reading DNA. It would be, and there were organisms that existed before DNA, right? So it doesn't seem so preposterous to me that we'd have intracellular mechanisms for reading RNA.
Oh, I'm not saying that we don't. I mean, obviously we do, in the sense that we synthesize proteins from RNA. The question is,
I mean,
how do those internal processes manifest externally in a way that affects the behavior of the organism in such a way that we're inclined to attribute memories to that organism?
Yeah, I mean, I guess this argument feels very abstract to me, because like if we accept that you read out things like you construct proteins, for example, from RNA, then what's to stop you from conjecturing that you read out other things that affect behavior? I guess what we need really is an empirical demonstration, right? That's kind of what I'm angling towards.
I mean, I don't think this is abstract at all. Like, if you believe something like the neuron doctrine, if you believe something like the main drivers of behavior, interestingly complex behavior, are neural activity, then there has to be some way that internal protein synthesis or RNA reading or whatever affects neural activity.
Right, I agree with that. Right, I just don't see what the problem is.
So it's only going to affect neural activity through spiking, say, or through synaptic modifications.
No, not necessarily. I mean, cells can send RNA to each other through vesicles.
That's very slow.
Yes, that's true.
And not very long distance.
That's true.
So how do you use something like that to drive behavior in a large organism, in an organism with like millions of neurons? Or,
well, I'm not saying it's the only driver of behavior, or even a typical.
But I'm saying
driver.
If it can't drive behavior at all, then why should we think that it counts as a memory?
Well, I didn't say that it can't drive behavior at all either. I mean, I think it can drive behavior, and we have empirical demonstrations that it can, in the sense that organisms can acquire taste aversions from transfer of RNA in these vesicles. But that's not. It's just. I think we're agreeing on everything, right? We agree that spiking activity and synapses are important for mediators of behavior. There's an unanswered empirical question about whether memories can be stored in RNA molecules and then read out in such a way that they can affect spiking activity. We don't exactly know that.
So there are some related comments by Niranjan Kambi that I really appreciated. One of them says, a single cell can carry information about its environment. It has, insofar as it can carry information about its environment, it has memory and behaves adaptively. And another time calls memory, she says, memory is carrying information forward in time and expressing it at a later time. That's also an interesting, I think, point where if you have some information and you retain it and use it later, I think it's fair, even if it's just the behavior that is shown in that kind of a context, I think it's fair to call it a memory, even if it's a single cell. Another thing but I think I appreciate always when Rosa does this. So Rosa is a philosopher, and every time she kind of questions these things, I think it sort of activates a certain sense of reevaluating. So maybe we will look back at this and say, okay, maybe we should call it something other than memory, or maybe we should dissociate between reading versus behaving, although in a single cell that's harder, I think, to dissociate. Another thing that Niranjan said that I'd like to, I think it's related, is it is thermodynamically expensive to store memories using spikes rather than thermodynamically stable molecules like DNA and RNA. And I think that in this comment that was going on in the chat, there's something interesting about a kind of the homeostasis of having memory, basically, and the kind of the costs that it might have for different kinds of organisms. And so I wonder what you think of that idea, Sam, that perhaps it is less expensive for fewer cell organisms or earlier organisms to store via RNA and DNA, and gradually, I think the kinds that Rosa was mentioning might become more representative of memory or other kinds of memory when you have enough cells and enough caloric intake, so that you can sustain this homeostasis of memories maintained with multiple cells.
Well, what exactly is meant here by memories with multiple cells? Because I think that's the critical question. Are we conceiving of memories as the synapses between the cells, or things stored in the cells?
So imagine spikes versus RNA, broadly construed.
Wait, but spikes are not memories.
Thinks that memories are stored in spikes.
What's that?
No, the spikes are not memories, though, right? They can't be, right? But spikes can convey information that's stored in memories.
That's what I have in mind. Yeah. So something that's emergent of multiple cells doing something, the way that we would think about memory stored in the human brain, for instance, about like an episode. It's not one cell that represents all of the memories. Mm-hmm. There is a number of cells together. There is an emergent pattern, and that sort of is read by somewhere else, whether I think about it or have a feeling about it or act on it or tell a story about it. it's a multi-cell sort of business. And it's not necessarily the case that, I don't know, your memory of your 7th birthday is transferred to your child. Like, that's not necessarily the case. So compare that to this RNA. I think what Niranjan was saying is that it is thermodynamically more expensive to have memories that are kind of mediated by spikes, mediated, let's say, by this kind of higher level multi-cell mechanism, as opposed to thermodynamically stable molecules like DNA and RNA. I hadn't thought about this before. I think it's this thermodynamic or like sort of homeostatic argument is interesting, especially when you have creatures with fewer cells.
Yeah. Well, yes. I mean, it's certainly more expensive to store information at synapses than in, say, a polynucleotide sequence. But I would want to clarify that that's not to the exclusion of the possibility that memories are distributed across multiple cells, right? Because you can have multiple cells store information in a polynucleotide sequence, but then for some complex memory, you have to have intercellular communication such that those pieces of information can get combined.
So that's a great point, and it was actually in one of the other questions. I don't know if I can find it. Someone asked about exactly multi-cell storage of them. Oh, here. Okay. Could facts be stored redundantly at a molecular level in multiple locations in multiple cells? That was Max did, Neil.
Yeah, I mean, I hope so. Otherwise, that would be bad news if the cell dies.
And then another related question by Anya. Would it imply that in a multicellular organism, non-neuronal cells have memories of their own? What would the consequences of this be? Do my liver cells remember my last Friday night? Maybe the immune system can be a good example for memories. Apart from the immune system, what is the use of these memories if there is no way to express behavior for a single cell in a multicellular organism? And I think that relates to Rosa's question kind of a little bit loosely.
But all right, so I mean, there's a bunch of things in that question. I think I just want to emphasize that we can talk about information being stored in a single cell in a molecular format, but to express that information behaviorally requires coordination between cells, intercellular communication, possibly that the form of that intercellular communication is mediated by synaptic plasticity. Indeed, that's quite likely. So it's, I'm not, when I say that memories are stored inside a cell, that doesn't mean, that shouldn't be taken to be the same thing as saying that like a single cell is expressing the memory in a way that we would recognize, right? Maybe for Paramecium that's true, right? But for multicellular organisms, it's unlikely to be true.
So Rosa just wrote something in the chat that I'd like to follow up on. Rosa says, and you can say it yourself, but I think the relevant question is, with regard to memory alloy, do you want to read that, Rosa, just so that, and I want to follow up on that.
I think some people in the chat were trying to make the case that memory alloys have memory or are able to learn, and it seems like that's actually a good example of why merely carrying information about the past is not enough to count as having a memory of the past, right? It seems like you need something more than that. You don't just need to carry information from some perspective, because like everything in the world carries information from some perspective about the past. Maybe this was also the point that Sam was making earlier about stars. It seems like you want something more than that. We want our term memory to pick out something more specific, more cognitive-y, something more tightly tied to living in the world and like behaving adaptively and learning from experience. So that means you have to be the kind of thing that can have experiences, the kind of thing that can use information that you've stored somewhere in your body somehow in order to behave adaptively in the future. And it seems like a memory alloy or a rock doesn't really satisfy those conditions.
Well, I think that memory, I mean, I would actually like to say that physical objects like alloys do have memory, but it's just a different it's not necessarily, as you say, it's not picking out the specific kinds of memories that are biologically relevant, potentially. Now they could be, right? Because if I, a biological organism, engineer a physical object to store memories on a behaviorally relevant timescale, then yes, I would say that those are storing memories
Those
that
are memories for
relevant.
us, yeah.
Right, yeah.
So on that,
But they're memories for you. It's like in virtue of your using them that they count as memories. And if like you went away, they wouldn't be memories anymore.
No, I think they still would be memories, but the timescales would be different. They would be dictated by the physical process rather than the biological organism.
I think I don't understand the last.
I mean, this is all a semantic argument, right? Because I'm taking memory to be a trace of the past, and you're taking it to be something more specific than that. I don't think we're going to be able to resolve that if we...
I don't think we need to resolve that. I think that's merely verbal. But I think in order for your thesis that single-celled memory organisms have memories to be interesting, and not just like obviously because every physical thing has memories, then you had better be talking about something a little more interesting.
Okay. Yeah, totally agree with that. Yeah.
So on that note, there are mushroom networks and sort of slime molds, etc., where it's not an individual that remembers something. It seems like the cognition is happening in a collection of individuals that are interconnected. And then together they might map something. They might remember the map of some, for instance, there was the Tokyo example, but it's not the individual. So the memory doesn't need to necessarily be read by the same, or the memory doesn't need to live entirely in one organism in that case. Would you, Rosa and Sam, still call that a memory, although it's not? It's necessary for all of those individuals to be a part of it, but if you remove any individual, the memory is not sort of contingent on any given individual. It's contingent on the collective maintaining something. Do we call that memory? Do we call that cognition? And if so, is it? Where do you draw that? Because it seems like for Rosa, it's really important that it's read and it's written and read and used in the same organism. But in the case of the mushroom network, it might not be any individual organism. So what do you guys think? Sam, Rosa?
Well, I'll say something quickly, which is I don't think I'm super committed to this idea, but this read, write, use thing I think is a Gallistel thing. So,
I see. So it wasn't. It was your perspective. Sorry, I thought that you were okay. So what
the second thing about the mushroom, about the fungus, is that I don't think it matters so much whether it's within a single organism or not. What we really want to know is, like, is it memory for the entity that we're interested in talking about having memories? So if you're interested in the whole population of fungi that are somehow like interlaced and functionally intertwined, and they all together use this information that they somehow store, then sure, say the collective has a memory. If you only care about individual fungus organism that maybe you can give a name to, although I think like maybe fungi are a bad example because it's really hard to draw biological individuality boundaries around those things anyway, then you have to ask whether a single organism can use it, a single one of those things, however you draw the line around the single one of those things.
Yeah. Yeah, I basically agree with what Rosa just said. I think that the questions we should be asking, at least the questions that I want to ask, are not about like, is X a memory, but rather I declare that I want to remember something, and I want to ask, what is the physical biological substrate of that memory? So if it's multi-cells or single cells, either could be a valid answer.
We have John here, and his internet is very flaky right now, so I want to just make sure we get an opportunity to hear what he has to say and hear his question before it stops working. So John, can you unmute yourself and ask a question?
We don't see him, or I don't.
Oh, really? I see him.
With your third eye?
Okay, yeah. Okay, well, I'll text him whenever he gets back in to just interrupt. It will be blunt and interrupty, but I think it's the only way we're going to actually get him to see.
Yeah, let's try that. Let's try that. We really miss John. Oh, something happened. I think there was a moment of flicker, like he left or something.
I'm calling in, so I'll put him on speakerphone and that'll work.
Oh, there he is.
He is.
Go ahead, John, before you lose connection.
Can you hear me?
Yes.
Can you hear me?
I can hear you. We can hear you, I think.
We will hear you again once you reconnect. It's a very pandemic moment.
Can I just say something about what I just said while we're waiting? So I think, I feel like what Sam and I just said is very much in the spirit of what you generally advocate, right? Which is like, let's not argue about the words, let's not argue about the boundaries around them, let's think about what's actually useful and what questions are actually at stake.
Yeah.
And like that seems like the right approach here as well.
Yeah. I think that in this moment, you and I switched places. You're usually the one who sort of asks about the memories, but I was more interested in your perspective in that sense of, if you're drawing such clear boundaries that this is not a memory, this is not a memory, this is a memory based on read and write and use, behavioral use in an individual, I wondered how far the limits of your position on that would be. So I was really more interested in your philosophical perspective to some extent. And so I'm glad to hear that in this case, you're functionalist like us, it seems. Awesome. Thank you. This was really fun. And while we're looking for John.
Okay, I have John on my cell phone on speaker. Let's see.
Awesome. Fantastic. That's the best approach.
John?
Yeah, can you hear me?
Yes.
Yeah, I'm sorry. I don't know why I'm having this trouble. It must be New Mexico. Can you hear me, Sam?
Yes.
Yeah, so, I mean, as always, I wish Rosa had joined the Salon more often in the past, because I find myself really sharing her inchoate concerns. I really enjoyed your article, by the way. I thought it was just, you have so many voices, Sam. It was historical, it was cultural, it was great, right? I mean, I really like the article this is all based on. But I can't help thinking of this all in part of a larger project, of sort of naturalizing memory and naturalizing representations by suddenly a mass infatuation everywhere with unicellular organisms, right? There's work on information processing in bacteria. The Carl Friston view applies just as well to bacteria as to primates. And now we've got learning in unicellular organisms. And it seems to me that there are two issues at stake here. One is Galastel's legitimate concern about synapses, right? And can they do the job that they've been given? And the other one is about how far down can one go and still use the word memory, right? And as you know, we're living in the world right now of vaccinations, when people talk about memory cells in the immune system, you can transfer immunity from a mother to a child. So we've been here before when it comes to the immune system. The question is it really true that these are all quantitatively the same thing? And qualitatively the same thing, or are they different things? Rosa mentioned the Tanagawa work, right, which sort of agreed with Galastel on one level, that actually the ability to form a memory optogenetically in an ensemble of cells, which they called an engram, was not in fact affected by the strength of synaptic connections. So, Thomas Ryan and the team actually thought kudos to Galastel, but actually the memory was in the configuration of the ensemble, which could be triggered optogenetically, but was not in fact related to the synaptic strengths. In fact, the synaptic strengths related, if I remember, only to the retrievability, not the quality of the memory, right? But nevertheless, to Rosa's point, those famous experiments by Wilder Penfield, when he would stimulate a particular location of the temporal lobe in a patient, and an incredibly vivid formed memory would come up, right? A corridor, a door, a person. So are we just playing a mnemonic game here and calling everything memory, from paramecium to immune systems to remembering a vivid taste of ice cream and a favorite friend, and you're just going to put them all under the same qualitative designation? Is that really what this is about?
John, can I jump in here? No, I think that you've already planted the seeds of an answer to your own question, which is that there's really two questions, which is this all memory? To which I'll say yes. But there's another question, which is, are these all the same kind of memory? To which I will say no. And I think that was basically the outcome of the dialogue with Rosa a few minutes ago, which was that, I mean, we might disagree about exactly what are the minimal conditions for memory, and I draw that boundary pretty broadly as basically anything that retains information about the past. So the universe can be a memory. But if I want to say, does the paramecium store a time interval and retrieve it later to guide behavior, right? That's a very specific kind of memory, right? So it's not the case that we say that, all right, anything could encode this memory, this particular kind of memory. I don't know if that satisfies you.
Yeah, can you still hear me?
Okay.
Yeah.
Yeah.
Yeah, but I'm just trying to understand what's at stake, right? In other words, that's what I'm trying to understand. In the information work that's being done, it's because they're looking for a universal mathematical theory of everything, right? In other words, they want some unifying principle that is thermodynamics, statistics, uncertainty, information, and that's the attraction. What's the attraction here? Is it the possibility that the synaptic story was wrong and it's actually inside cells? Or are you trying to come up with a universal theory of memory, starting with the simplest?
No.
I don't know what the project is.
Okay, yeah. So sorry, and unfortunately you missed my talk, and maybe that would have been clarifying. But my project is
the project.
My project is very much not coming up with a universal theory of memory. I mean, I'd be certainly interested if someone wanted to write that, and I would read it. But my project is to explain specific kinds of memory in specific organisms, right? And I want to understand, first of all, does this organism, does a paramecium store memory about, for example, time intervals or the information theoretic contingency between events? And then if I can provide behavioral evidence for that, then I would want to look for the molecular substrate of that and the read and write operations that act upon it, right? That's my project.
So in other words, this will be the same as being interested in sound localization in bats. You're just particularly interested in an idiosyncratic
No.
behavior. So what induction are you hoping to happen in the background?
All right, good. Yeah, so no, I'm not interested in this specific idiosyncratic process. I mean, I think it's interesting to understand for its own sake, but my longer term agenda here is to provide a proof of principle for how it could work, how memory could work in a non-synaptic way that could potentially be generalized to other organisms and other forms of memory. That's rendered plausible by the fact that Paramecia share a lot of their intracellular biology with much more complex organisms. First we have to start with the Paramecia.
But so that does imply that the sort of Tonegawa N-gram Wilder Penfield sort of, Hopfieldian, you get in one part and you can light up the whole network, sort of N-gram notion. Are you saying something contrary to the N-gram notion, or are you thinking about how it might be implemented? Is this an implementational version of the N-gram story, or is it an alternative to the N-gram story?
No, I don't think it's contrary to the existence of N-grams, or it's not contrary to the existence of cells that report a memory, right? But that's not saying very much, I think, about this specific implementation. So yes, it's really a question about implementation. The way that I see it is that you could potentially store information at the level of single cells, but then what we would describe as a memory unit, like a memory of an episode, for example, is distributed across multiple cells. There's not one cell that encodes all the information about that episode, and therefore retrieval of that memory entails the co-activation of multiple cells.
But there's a little bit of a difference between a memory being in the configuration of all the cells together, like Thomas would argue, versus each cell contributing its little bit of micro memory, and then the total memory is just the sum of the micro memory. So it was a strawberry flavored ice cream in one cell, and it was a cone in another cell. I mean, is that what you're saying?
Well, okay, so I mean, I don't want to put words in Thomas's mouth.
It's the network. Which one is that?
Yeah, I don't want to put words in Thomas's mouth, but I'm pretty sure that he would disavow the idea that information is stored intracellularly, right? So in his view, the memory is stored in the connections, and I think more recently he's entertained the idea that there's a kind of structural connectome that mediates the memory engram. But I think that I don't believe necessarily that the kinds of the information stored at the level of a single cell has to be symbolically identifiable with things like cone or strawberry, right? In much in the same way that when we store information in digital computers, we don't necessarily have to be able to interpret the bit patterns in that way, right? But we could talk about distributed storage of information that then gets assembled, and there's an interface at which we can then read off the assembled information, right? That's, I think, what they're measuring when they measure the n-gram cells.
So would it be fair to say maybe the reason we're struggling here is that it's the old thing between procedural versus declarative memory, and that you're showing evidence of procedural like memory and savings and other such things in a paramecium, and computers can do procedural memory, and what we're all maybe sticking on is this notion of declarative memories, these overt image-based symbolic maybe. Is that what this is about? That we're really having a discussion here about intracellular procedural memory could happen, especially if it's paramecium, and that what we're really not so easily translating to is how would we get what we call declarative episodic memory out of what you're describing inside the paramecia?
I mean, I'm personally not so eager to adopt that distinction, because even the declarative memories have to have some realization at the physical level in cells, either in the synapses or inside the cell, or both. So if we want to say that something's procedural because it involves I guess I'm just not totally understanding what you mean, what your definition of procedural is, right? Because the traditional procedural declarative distinction is really at the level of behavior, right? It's taking out certain classes of memories that can be measured in different ways. But what does that mean at the cellular level? They could both be the same. They could both be mediated by the same molecular substrates at the cellular level.
Well, actually, you're right. I remember having this discussion with Kandel, right? I mean, LTP and LTD and all the kinases are present everywhere. So where does the difference come? In other words, aren't you hoisting yourself by your own petard? But if you say that, then where does the difference come from? In other words, if the cells can basically do it, where does the actual difference come from? For example, between the cerebellum and the hippocampus. In other words, once you dive down at the level of the cells and the processes and what they can do, it's all the same. Where does the difference come from?
Right. I don't know. It's a great question.
I mean, I feel like you just said it, right? The difference comes from what parts of the brain are doing with those differences at the cellular level.
Well, where does it cash out? That's what I'm asking. Is it at the ensemble level because the cells are all pluripotent? Where does the divergence happen in your view?
Well, I mean, your computer has different stuff on it than my computer, probably. So, I mean, but it's made out of the same stuff. The circuits are the same, right? So it's not. It's easy to imagine how you can build circuits neural circuits out of the same stuff, but then store different information on them. I mean, I don't think that's entirely correct, though, because
I'm being a bit obtuse here. I mean, there must be something about using the same stuff. You can build a church or a hotel out of the same bricks, or you can build a dome or a spire out of the same bricks. But a dome and a spire are very different things, right? Even though they've been made out of the same stuff. So there must be a point where the difference manifests itself between the two different types of memory, just like a dome is different from a spire, don't you think?
I mean, look at biology, right? We have stem cells that can differentiate into all sorts of different kinds of cells. How do they do that, right? A lot of it has to do with how their external environment affects them. But I mean, I don't know if it's a very profound statement.
Emergent,
yeah,
that we made out of the same stuff that we're kind of getting rid of by going too basic, right? But maybe that's just a perception.
I mean, incidentally, the cerebellum and the cortex are not the same stuff, right? I mean, the cerebellum has cells that are quite remarkably different. So the case could be made that they're not the same stuff.
Right, the architecture, the macro architecture is different. But if you go inside there, I don't know the answer to this. They may have the same kinases. They may have the same RNA readers, right? I mean, So at some level, from your argument, they should be pluripotent, right? But then at the same level, there must be something about their architecture and the way they're hooked up with everything else that makes them do a very different job, even though they may share kinases and RNA readers.
Mm-hmm.
Do you see what I'm saying?
Yeah, I think so. But I guess I mean, I don't know if anyone else wants to jump in here. I could keep responding.
I'm just trying to understand what the similarities turn into differences that matter.
Yeah, I think, let me see if I understand what John's saying. I mean, Sam, to your point, clearly, I'm happy with your definition. Everything is a memory, basically, and that there's lots of different kinds. And I think John's point is, which kinds do we want to understand, and how do they relate to one another if we're going to try to understand human cognition and how to build artificial intelligence? And is this one of those kinds? And if so, how is it related to the other kinds? Is that right, John?
I was just saying that I was just under the impression that at certain points, the configurations of cells, despite their internal similarity or identical nature, begins to qualitatively change what the ensemble does in the memory space, even though what each cell might have inside it is very similar. There must be a point where even though you're made of bricks, you become a church rather than a hotel. And that seems to be at the macro level. And if it's at the macro level, then I don't see how studying what's happening inside a single cell is going to get us there.
Yeah,
it could contribute, presumably. Right, you want to know how to make the bricks if you want to make either a church or a hotel. So if Sam is telling us something useful about how some of the constituents of the memory maybe are, created inside single cells, then it seems like that would be an important contribution.
Yeah, I'm not saying that you take a bunch of Paramecia and put them together and you have a brain, right? I'm not making that claim. I'm just saying that some things might be conserved across evolution.
So if I may bring this to the kind of the usually late hour discussions that always comes back to it, I think that there might be a kind of a paradigmatic difference between what John wants to consider cognition or representations and what some of us who do computational work might be comfortable calling that. I think that there is a kind of a preference, and John, please correct me if I'm wrong. I think there's a preference for John to theoretically ascribe the notion of even representation memory to things that are much more higher level in terms of cognition than what would happen in, for instance, sort of not so high level organisms. And this has been a recurring thing that in many conversations comes up. And I wonder whether the difference right now is based on that, whether John would be comfortable with a single cell having a memory, or a very sort of few cell organism having a representation, basically. And maybe the question John was asking earlier, I wonder whether that was the kind of the paradigmatic higher level framework in mind when he was asking that. John, was that a reasonable capturing of potential differences here?
I definitely do have as you say, probably a more restricted notion of what we should call cognition and memory, a little bit like the way Rosa was saying. But actually, this is a little bit orthogonal to that. I'm trying to understand why it is that, at some point, you may share internal cellular memory machinery and mechanisms to read the RNA and transfer it. But at some point, the way you organize those cells, just like Sam said, you would just put a bunch of paramecium together and suddenly you have a multicellular organism. There must be some qualitative emergent extra feature to the memory that comes from the configuration and the connections between them. And if that's the type of memory that comes out of multicellular configurations, it might just be a qualitatively different beast to the kind of simpler memory that is happening inside a paramecium. That's my only concern, is that they're not really intellectual friends. They're probably false friends.
Well, okay.
And they're friends because of the word memory sort of linking them. But, that's a little bit like saying that music and architecture and painting are all art. Well, yes, they're all art, but it's not particularly useful to the actual job of composing music or making a sculpture or building a building. The fact that they're all art isn't going to do much of the work after you've made that generalization.
John, let me make the case that they are true friends. And by analogy with computer engineering, we know how to build very complex computing devices out of the combination of relatively homogeneous and very simple components. And so it doesn't seem so implausible to me that you can get complex memories as an emergent property of putting together lots of simple computing devices. Like if a paramecium can store a time interval, and maybe another paramecium can store, the location of a food, and another paramecium can store the identity of the food. Now we have the rudiments of a more complex memory that's like what, where, and when, right? Those are the minimal criteria for episodic memory.
And in other words, Sam, you have kind of, and I'm glad you said that, but then you have got a bigger project. You actually do think that from this little building block of the paramecium, you can get something a lot more impressive. So you're being a little bit disingenuous when you said you weren't trying to build something bigger, which is great. Right?
No. I'm trying to.
I just feel that it doesn't quite work for me. The building block idea. It just doesn't. It just feels under-conceptualized. I mean,
yes.
That happens all the time in neuroscience. Well, let's do the building block behavior or the building block organism, and by some magical extrapolation, we'll get to the really fancy stuff. I'd like it to be a bit more fleshed out than that, and that the only flesh really is that you can use the word memory in both cases. That doesn't seem. You're going to have to do more than that, wouldn't you agree?
Yeah. I mean, I don't think we're going to answer this, right? Because it's so speculative. But whether or not, that bigger project is successful, I can still try to answer the questions like, does a single cell encode some information about, for example, contingencies between two events? And that doesn't necessarily bear upon the larger project of like putting a bunch of these things together makes an episodic memory.
Yeah, I just think you are oscillating a little Nepotube-like between just being local and modest and using the word memory to secretly, as you just revealed, thinking that you can do some kind of extrapolation to something.
No. John.
But it's just that you're oscillating a bit, and if you're going to oscillate towards the grander one, I'd like to hear a little bit more than just, well, it's kind of a memory.
Well, John, I think that what you're seeing as oscillation is just like my approach is to try to start with claims that I can actually make on the basis of empirical data, and then I'm not going to simply extrapolate that to multicellular organisms. That would be a huge mistake. If I wanted to demonstrate, all right, let's suppose the following. Suppose that I was able to establish that Paramecia
You're breaking up to me, Sam. It might be me. I know I'm just not hearing. I'm losing my words. Is it you, Jobo?
I can hear him. I think it's not me. It's you, John. Sorry.
All right, I'll try to be quick. Like, suppose that I could establish that.
Sam, sorry.
All right, suppose that I could establish that Paramecia store memories about contingency or time in a polynucleotide sequence like RNA. And I could not just establish that, but I could establish exactly what the code book was, how they're encoded in the polynucleotide sequence, how they're written, and how they're read. Okay, so now I have a complete story going from inputs to outputs in that single cell. Now the question would be, now that I've understood those mechanisms in this simple organism, can I then test whether those same mechanisms happen in more complex organisms, right? Do they mediate memory storage and retrieval in more complex organisms, right? So that's another empirical project that requires its own data collection, right? Not extrapolation.
John, are you with us?
Yeah, I'm here.
Can I add something to that? So when I think about sort of computational ideas of learning or computational modeling of things, the same way that, social things become as real as biological things, in the same way, memory in a single cell is as real as in a multi-cell organism, because when you're considering a model of them, then they're as real as each other in a way. So I think that might be a philosophical difference in this case. It's not necessarily the case that because all of them are computationally capturable, one is necessarily the building block of the other, but there is a connection. And for me, memory and learning in all of them is as real, whether it's at the social level with a collective of mushroom individual fungus individuals, whether it's in one individual that is a mammal and is remembering event, or whether it's in a single cell that's learning something via its RNA or other kind of ways cellularly. And I think that in that kind of computational way of thinking about learning and memory, I personally think it's a great contribution that Sam is doing to revisit these kinds of single cell learning ideas, because when we are thinking about the computational models of learning and memory, it could functionally, again, functionalistically speaking, it could capture all of these things. Are there differences? Absolutely. The same way that you can have different algorithms for learning, different types of memory. But I think it's a huge contribution to note that this similar sort of computational ideas or principles of learning and memory might be underlying single cells. A lot of sort of realization level differences, but algorithmically speaking, there might be some similarities. And I think that's a huge thing that I take away from this, but maybe I'm reading too much into it. Sam, do you want to say something about that?
Sorry, I was just turning to mute because there's a lot of...
I have to drive to Saturday. I want to thank Sam. Thank you so much, and I hope we can catch up. It's fascinating stuff. But I hope you know that just giving you a hard time because that's what we're meant to do at the Salon.
John, I wouldn't have it any other way, John. Thank you.
We love you, John.
And also, Jobo and Ida, thank you for hunting me down. That was very kind of you. You could have just forgotten about me.
Never, John.
The rest of the conversation as well. Big hugs.
Thank you.
Never without you, John.
Bye. Bye.
Go on, Sam. By the way, we always welcome kids here, but I understand that you want to go and sort of hang out with the kids. So if you want to say something to that or some final things, please feel free.
I mean, yeah, I'm gonna have to go in a few minutes just to have dinner with my kids. But I've lost my train of thought. I mean, I think that what you were saying made sense to me. and it kind of recapitulates some of the themes that we've been discussing.
Right. So the computational principle, making it as real across them.
Yeah.
I personally enjoyed this very much. A lot of people in the chat were saying that this should be on one of the, some people say top 10, some people say top 3 salons of all time. The chat has been pretty awesome. I don't know how much of it you've managed to see. My favorite was that at some point Elle said, yes, which is why I wouldn't say the bike has memories. And I think out of context, that's a fantastic banner for today's conversations. Thanks again. This was fantastic. And just a little thing, you're going to have a book coming out soon. You want to tell us like one sentence or two about that?
Oh, well, the book has nothing to do with anything that we've been talking about, really. I mean, not directly, at least. It's about human intelligence. It's called What Makes Us Smart. And it's sort of the thrust of the book is about trying to understand the various weird and seemingly irrational things we do through the lens of computational models of cognition.
Awesome. So maybe next semester we'll have you back with the book.
Sure.
Thanks again.
Yeah, thanks so much for having me. This was a lot of fun.
With the dinner. Yeah, it was so much fun.
Thanks, Sam. Thanks, Rosa. Thanks, Ida. Thanks, John. Thanks, everyone.
Thank you, Claire, and thank you, Bahar, for behind-the-scenes activities that sustain us.
And Worldwide Neuro.
Yes. Until next week. Bye-bye.